Why sex becomes less satisfying with age. The number of women regularly having sex declines with age, and the number of women enjoying sex post-menopause is even lower. Although these facts are not surprising, the causes for these declines may be because previous research focused largely on biological causes only. However, a new UK study identifies psychosocial contributors. Study results are published online today in Menopause, the Journal of the North American Menopause Society, NAMS. It's hard to pick up a woman's magazine or OB/GYN journal anymore without reading an article about how and why a woman's libido and level of sexual satisfaction decline during and after menopause. Substantial research has been conducted into biological reasons such as hot flashes, sleep disruption, vaginal dryness, and painful intercourse. Much less is known about the effect of various psychosocial changes that are common post-menopause. These include body image concerns, self-confidence and perceived desirability, stress, mood changes, and relationship issues. Of the research that has been conducted regarding psychological influences, most of it has focused on quantitative results. A study of nearly 4,500 postmenopausal women involved in the UK Collaborative Trial of Ovarian Cancer Screening (UKCTOCS), however, looked at free text data to better understand why women felt a certain way and the depth of those feelings. Among other things, the UKCTOCS sexual activity data showed that, at baseline, before the start of annual screening, approximately half of the women were sexually active. A decrease in all aspects of sexual activity was observed over time. Sexual activity was less frequent, not as pleasurable, and more uncomfortable. The primary reason for absence of sexual activity was the lack of a partner, mainly because of widowhood. Other commonly cited reasons for decreased activity included, in rank order, a partner's medical condition, a partner's sexual dysfunction, the woman's own physical health problems, menopause-related symptoms, and prescribed medication. Contributing most often to low libido were relationship problems, logistics, and perceptions of aging. Only 3% of participants described positive sexual experiences, whereas only 6% sought medical help for sexual problems. Study results appear in the article, Sexual Functioning in 4,418 Postmenopausal Women Participating in UKCTOCS, a qualitative free text analysis. Sexual health challenges are common in women as they age, and partner factors play a prominent role in women's sexual activity and satisfaction, including the lack of a partner, sexual dysfunction of a partner, poor physical health of a partner, and relationship issues, says Dr. Stephanie Faubion, NAMS medical director. In addition, menopause-related problems such as vaginal dryness and pain with sex have been identified as problems affecting sexual function, yet few women seek treatment for these issues, despite the availability of effective therapies. New cause of cell aging. Discovered new research from the USC Viterbi School of Engineering could be key to our understanding of how the aging process works. The findings potentially pave the way for better cancer treatments and revolutionary new drugs that could vastly improve human health in the twilight years. The work, from Assistant Professor of Chemical Engineering and Materials Science Nick Graham and his team in collaboration with Scott Fraser, Provost Professor of Biological Sciences and Biomedical Engineering, and Pin Wang, Zorab A. Caprillion Fellow in Engineering, was recently published in the Journal of Biological Chemistry. To drink from the fountain of youth, you have to figure out where the fountain of youth is, and understand what the fountain of youth is doing, Graham said. We're doing the opposite, we're trying to study the reasons cells age, so that we might be able to design treatments for better aging. What causes cells to age? To achieve this, lead author Alireza Delfera, a graduate student in the Graham Lab, focused on senescence, a natural process in which cells permanently stop creating new cells. This process is one of the key causes of age-related decline, manifesting in diseases such as arthritis, osteoporosis and heart disease. Senescent cells are effectively the opposite of stem cells, which have an unlimited potential for self-renewal or division, Delfera said. Senescent cells can never divide again. It's an irreversible state of cell cycle arrest. The research team discovered that the aging, senescent cells stopped producing a class of chemicals called nucleotides, which are the building blocks of DNA. When they took young cells and forced them to stop producing nucleotides, they became senescent, or aged. This means that the production of nucleotides is essential to keep cells young, Del Ferra said. It also means that if we could prevent cells from losing nucleotide synthesis, the cells might age more slowly. Graham's team examined young cells that were proliferating robustly and fed them molecules labeled with stable isotopes of carbon, in order to trace how the nutrients consumed by a cell were processed into different biochemical pathways. Scott Fraser and his lab worked with the research team to develop 3D imagery of the results. The images unexpectedly revealed that senescent cells often have two nuclei, and that they do not synthesize DNA. Before now, senescence has primarily been studied in cells known as fibroblasts, the most common cells that comprise the connective tissue in animals. Graham's team is instead focusing on how senescence occurs in epithelial cells, the cells that line the surfaces of the organs and structures in the body, and the type of cells in which most cancers arise. Graham said that senescence is most widely known as the body's protective barrier against cancer. When cells sustain damage that could be at risk of developing into cancer, they enter into senescence and stop proliferating so that the cancer does not develop and spread. Sometimes people talk about senescence as a double-edged sword, that it protects against cancer, and that's a good thing, Graham said. But then it also promotes aging and diseases like diabetes, cardiac dysfunction or atherosclerosis and general tissue dysfunction, he said. Graham said the goal was not to completely prevent senescence, because that might unleash cancer cells. But then on the other hand, we would like to find a way to remove senescent cells to promote healthy aging and better function, he said. Graham said that the team's research has applications in the emerging field of senolytics, the development of drugs that may be able to eliminate aging cells. He said that human clinical trials are still in early stages, but studies with mice have shown that by eliminating senescent cells, mice age better, with a more productive lifespan. They can take a mouse that's aging and diminishing in function, treat it with senolytic drugs to eliminate the senescent cells, and the mouse is rejuvenated. If anything, it's these senolytic drugs that are the fountain of youth, Graham said. He added that in order for successful senolytic drugs to be designed, it was important to identify what is unique about senescent cells, so that drugs won't affect the normal, non-senescent cells. That's where we're coming in, studying senescent cell metabolism and trying to figure out how the senescent cells are unique, so that you could design targeted therapeutics around these metabolic pathways, Graham said. High levels of estrogen in the womb linked to autism. Scientists have identified a link between exposure to high levels of estrogen sex hormones in the womb and the likelihood of developing autism. The findings are published today in the journal Molecular Psychiatry. The discovery adds further evidence to support the prenatal sex steroid theory of autism first proposed 20 years ago. In 2015, a team of scientists at the University of Cambridge and the State Serum Institute in Denmark measured the levels of 4 prenatal steroid hormones, including 2 known as androgens, in the amniotic fluid in the womb and discovered that they were higher in male fetuses who later developed autism. These androgens are produced in higher quantities in male than in female fetuses on average, so might also explain why autism occurs more often in boys. They are also known to masculinize parts of the brain, and to have effects on the number of connections between brain cells. Today, the same scientists have built on their previous findings by testing the amniotic fluid samples from the same 98 individuals sampled from the Danish Biobank, which has collected amniotic samples from over 100,000 pregnancies, but this time looking at another set of prenatal sex steroid hormones called estrogens. This is an important next step because some of the hormones previously studied are directly converted into estrogens. All 4 estrogens were significantly elevated, on average, in the 98 fetuses who later developed autism, compared to the 177 fetuses who did not. High levels of prenatal estrogens were even more predictive of likelihood of autism than were high levels of prenatal androgens, such as testosterone. Contrary to popular belief that associates estrogens with feminization, prenatal estrogens have effects on brain growth and also masculinize the brain in many mammals. Professor Simon Baron-Cohen, director of the Autism Research Center at the University of Cambridge, who led this study and who first proposed the prenatal sex steroid theory of autism, said, "This new finding supports the idea that increased prenatal sex steroid hormones are one of the potential causes for the condition. Genetics is well established as another, and these hormones likely interact with genetic factors to affect the developing fetal brain." Alex Sumpanidis, a PhD student in Cambridge who worked on the study, said, "These elevated hormones could be coming from the mother, the baby or the placenta. Our next step should be to study all these possible sources and how they interact during pregnancy." Dr. Alexa Pohl, part of the Cambridge team, said, "This finding is exciting because the role of estrogens in autism has hardly been studied, and we hope that we can learn more about how they contribute to fetal brain development in further experiments. We still need to see whether the same result holds true in autistic females." However, the team cautioned that these findings cannot and should not be used to screen for autism. "We are interested in understanding autism, not preventing it," added Professor Baron Cohen. Dr. Arya Cohen, the biochemist on the team, based at the State Serum Institute in Copenhagen, said, "This is a terrific example of how a unique biobank set up 40 years ago is still reaping scientific fruit today in unimagined ways, through international collaboration." The research was supported by the Autism Research Trust, the Medical Research Council, and Wellcome.